we have entered the small intestine. Let's get an overview of the small intestine first. It is divided into 3 subdivisions. The first section of the small intestine is the duodenum. Some people also pronounce it duodenum, so tomato, tomato. The duodenum is the shortest part of the intestine. It's about 25 centimeters long, and it has this nice little curve here, nestled up with the liver, and then the head of the pancreas kind of is also nestled into the curve of the duodenum. The next region of the small intestine is the jejunum. It's about 2.5 meters long. And then the last region of the intestine is the ileum, which is about 3.6 meters long. So we have lots of intestine all squished up in there. Now, the small intestine is the major site for digestion and absorption. So far in the gastrointestinal tract, we have talked about how there was a little bit of digestion that took place in the mouth, a little bit of protein digestion that took place in the stomach, a little bit of absorption just of alcohol from the stomach. But the vast majority of chemical breakdown, of digestion, as well as absorption of nutrients from the lumen into our bloodstream is going to be taking place in the small intestine. The small intestine is also going to be involved in mechanical breakdown through segmentation. So that's kind of the squishing back and forth. And then additionally, in order for this food to get all the way through our intestine, the small intestine is also involved in propulsion through peristalsis. So that's the movement like pushing toothpaste through a tube. So let's take a little closer look at the structure of the small intestine. So here is that picture again, that cross section of the small intestine that we use to look at the different layers that make up the gastrointestinal tract. And I want to point us let's take a look at this mucosal layer. And you can see how there are these finger-like projections, lots and lots of them, finger-like projections towards the center of the lumen. These finger-like projections are called villi. Villi plural, villus for singular. So what these villi are doing is they are increasing the surface area of the mucosal layer. And that is especially important because one of the big functions of the small intestine is to absorb nutrients from the lumen across the epithelial cells so they can finally get into our bloodstream. So in order to efficiently absorb nutrients, we need lots and lots of surface area. So that's what we see here. These are the villi. And then here is a zoomed in view of one villus. And what we can see is that this one villus is formed of many cells. These are called enterocytes. They're all lined up in a row. And then if we look inside this villus, we can see that we have some blood. If we look inside this villus, we can see that we have some blood vessels. So what will happen is nutrients will get absorbed. The nutrients will get absorbed on this side of the enterocyte, across the enterocyte, and then go straight into the blood vessels. We also see in green that we have this nice lacteal, and that is the type of vessel that gets drained into the lymphatic system. Now let's take a closer look at the individual cells that are lining this villus. So this is an individual enterocyte, and we can see that this enterocyte also has a specific structure, these kind of hair-like projections, and these are called microvilli. So these microvilli, again, are going to be serving the same purpose to increase the surface area of this particular enterocyte so it can be more efficient at absorbing nutrients. I have always thought that these microvilli on the enterocyte look exactly like Bart Simpson's hair. So if it helps you remember that, I usually think of enterocytes as Bart Simpson cells, but enterocyte's their actual name. Okay, so let's take a closer look at these enterocytes now to see how the enterocytes kind of finish up the digestion, so the chemical breakdown of our macronutrients, and then the enterocytes are absolutely critical for the absorption of these nutrients. So in this diagram, I am showing you that there are some enzymes that are lining the tips of the microvilli of our enterocytes. And these are enzymes that are going to be involved in some of the final digestion of our macronutrients. So we'll have enzymes here that are important for digesting short peptides into either individual amino acids or di or tripeptides, kind of two amino acids strung together or 3 amino acids strung together. So that's some of the final digestion of proteins that will happen here. Same thing for carbohydrates. We will have some enzymes that are lining the tips of the microvilli that are going to be involved in digesting disaccharides down into monosaccharides so that we can absorb them. Now, we also, so I mentioned that the enterocytes are critical for absorption. Now, first, I want you to think about this concept, that in order for a nutrient to get from the lumen of the intestine into our bloodstream, that nutrient is going to first have to cross the luminal side of the enterocyte, and then it's going to have to leave out the back door across the basolateral side of the enterocyte. Represented in this image here, I am showing you several different kinds of transporters on the luminal side of the enterocyte, and then several other kinds of transporters on the basolateral side of the enterocyte. As we move through the semester, we will talk in very specific detail about what are the transporters required for the luminal side for specific carbohydrates, specific proteins, what are the transporters that are required on the basolateral side for specific carbohydrates, for specific proteins. Once a nutrient has gone through the luminal side, traveled through the enterocyte, and then exited out the basolateral side, then it can enter into the bloodstream, and for the proteins and the carbohydrates, it will go straight to the liver. The liver gets the first pass of those nutrients. Things are a little bit different when it comes to fats. So fats, we already know they are going to be emulsified with the bile salts. Once the bile salts have helped emulsify the lipid droplets into smaller lipid droplets and our lipase enzymes have broken down the fats, the digestive fats, which will now be in their small units of fatty acids, cholesterol, as well as fat-soluble vitamins, they are going to get packaged up into something called a micelle. And so that's what I'm representing here. These are the micelles. I blew up a micelle over here so we can look at it in a little bit more detail. We can see that this micelle is composed of a whole bunch of phospholipids. Now, what we remember about the phospholipids is they have this hydrophilic phosphate head, and then they have these two fatty acid tails. And so this is a very comfortable arrangement for our hydrophobic lipid, digested lipid compounds, the fatty acids and so forth. So the fatty acids are going to be surrounded by the fatty acid tails of the phospholipids, and then the phosphate heads are going to be oriented towards the outside in the watery environment. So what happens here is we now have these micelles that are full of our digested lipid. This micelle package is then going to just diffuse across the enterocyte, diffuse right through the membrane of the enterocyte to get inside the enterocyte. Once our digested lipid components are inside the enterocyte, they're going to get repackaged up in another form called a chylomicron. And from there, that chylomicron is going to diffuse across the basolateral side and actually enter into the lacteal and enter into the lymphatic system. So we'll talk in much more detail about this when we get to the lipid section of our course. But for the time being, what I would like you to appreciate is that the lipids that we have digested from our food, they are first going to enter the lymphatic system. And so they're actually going to skip that first pass of the liver. So it's different from the proteins and carbohydrates that we digest and absorb. Okay, we have now traveled through the entire small intestine. We have arrived at the large intestine. The large intestine is mostly going to be involved in absorbing water. But the other nutrients have primarily already been absorbed in the small intestine. So the large intestine will be absorbing water. We know what happens if the large intestine does not absorb enough water. That's when you're going to have a situation with diarrhea. The large intestine is also involved in absorbing some vitamins and some electrolytes. There's no digestion that's going to be going on in the large intestine. All of the digestion has already taken place in the small intestines. And I'm just going to clarify that a little bit by saying there's going to be no digestion by human enzymes that is going to be taking place in the large intestine. More on that in a second. The large intestine is also going to be involved in propulsion. So again, squishing to move the remaining undigested or the remaining food excrement through the large intestine. And then finally, of course, involved in defecation of feces. Now, the large intestine is rich with microbes, and this whole population of microbes, we call them the microbiota. And the microbiota, they can actually continue to digest and break down some of the components of our food that we couldn't break down. So that goes for some components like fiber, for example. So when I say there's no digestion that goes on in the large intestine, I'm really meaning no digestion by human enzymes, but there could be some additional metabolism by the microbiota. Now over here, I just want to point you towards this picture. This is a cross section of the large intestine. And what I'd like you to notice is that this mucosal layer is much thinner than when we were looking at the small intestine before. And additionally, the villi are much shorter. And that's really kind of giving us a sense of what's important in the small intestine. We knew that absorption of nutrients was really important in the small intestine, but now we don't have to do as much absorption in the large intestine. So that mucosal layer that's really responsible for absorption is much thinner. We don't need as much surface area there.